Hi, welcome. Let's move on to the sliding filament mechanism, but more specifically the chemical events that go on. In this diagram, you're seeing actin and myosin, but this muscle is relaxed. So myosin is not attached onto the actin. You can see how there's a space there. Tropomyosin is blocking the location of attachment for myosin, and nothing is happening. Okay, so we're at rest here. What changes? Well, the first thing that needs to happen is calcium levels need to increase inside the cell. So there needs to be a release of calcium from its storage location. And now I have these little balls of calcium. And the step that's going to happen then is that calcium is going to attach onto troponin. But let's take it one step at a time. The first one is calcium arrives. 2, the calcium binds to the troponin. So there you see the little ball onto the troponin. Next, this troponin complex, the troponin attached onto the calcium now, is a molecule that's going to change shape a little bit because now it has calcium attached onto it. And it's going to move. And in moving, it's going to drag with it the tropomyosin. So by dragging the tropomyosin out of the way, it's going to expose the binding site for myosin. And as you notice here, myosin can now attach because tropomyosin has been moved out of the way. And this head is sort of in that cocked position. What's going to happen next is that it's going to complete a power stroke. So this direction of the arrow is showing that it's going to move this way, and I'm going to have actin sliding in that direction. It'll then release and attach onto the next one and push, and release and attach and push, and release and attach. And that's what we're calling the power stroke. This diagram is showing how actin filament is going to move. Interesting to look at a rigor mortis. So my little dead stick man, it cannot release actin. Myosin won't be able to release its actin until a new ATP molecule binds. So I'm going to take you back to the previous slide. I need ATP both for attaching onto this in the power stroke and also for detaching. And in a body, in a corpse, if you run out of ATP, then you would not be able to remove this myosin head from attachment, and it would stay in a contracted muscle state. So you run out of ATP at death. Of course, you're not making any more of it. And those cross bridges between myosin and actin never release. That is what rigor mortis is, the stiffness of a body. Think when I show you this, I only show you one myosin head attaching onto the actin. But really you have to think of many of these cycles occurring at the same time. Lots of these golf club heads are coming up to attach and push and detach and attach and push. So many of them are occurring, and think they are occurring asynchronously. And that's an important word here. It's asynchronous. Think of what would happen if it wasn't asynchronous, if they all did it at the same time. If they all came up and attached and let go, it would slip backwards. So they have to, while one is attaching and pushing, the other one's letting go, and then another one will attach. And at any one time, there is some myosin attached onto the actin as it makes that actin slide. You also need a steady supply of ATP. And remember the role of calcium. If you don't have calcium, none of this can happen. You need that calcium coming into that region of the actin and myosin within the muscle cell. How does it get there? How does calcium get into the cytoplasm? Well, we store it in these cells, in your muscle cells. You have a huge storage unit, and that is the sarcoplasmic reticulum. So in this diagram, it's all in blue here. It's a network that's going to surround all these myofibrils. So it's very close to where it needs to be used. And anytime that action potential is coming from a neuron, then it's going to stimulate the release of that calcium from the sarcoplasmic reticulum. This is just an enlargement, and imagine a neuron coming down like this. And this neuron has stimulated an action potential that is going to come down into the inside through this T-tubule, and stimulate the sarcoplasmic reticulum then to release calcium to all of this area in order to start a contraction. This would be the surface, the sarcolemma. A T-tubule then is the entryway, is the place where that action potential can depolarize, come and depolarize the sarcoplasmic reticulum, and that is the trigger for releasing calcium to that actin and myosin area. If you look at it in the very big picture, you want to know what happens from the time the motor neuron comes to the very bottom here when the myosin attaches onto the actin. So there's lots of steps involved there, and you really need to know this slide. You really need to understand this. This is in the book. So let's take it one step at a time. A somatic motor neuron releases acetylcholine at the neuromuscular junction. Remember, right here, my little squares of acetylcholine. And it initiates an action potential on the surface of the cell. So there it is in red. This action potential is coming down this entryway, which is the T-tubule. And if it triggers this receptor here, then this receptor, it's called a DHP receptor, it is going to link directly to the sarcoplasmic reticulum and release calcium. Return this video if you need to listen to that again, because this is very important. What happens next? My action potential arrived there at that gate at the DHP receptor that turns out is linked with the sarcoplasmic reticulum. So the DHP receptor opens the calcium channels here in the sarcoplasmic reticulum, and guess what? Releases all these little green balls here that are the calcium. Now that is floating around the cytoplasm of the cell. The calcium next will bind to this little yellow ball, remember the name of that? Troponin or troponin, and trigger the removal of the tropomyosin ribbon. If I remove the blockage of the tropomyosin ribbon, then the myosin filament can attach onto actin and move it, and attach onto actin and move it, and attach onto actin and move it. And there I have the sliding mechanism happening. So this muscle contracts as long as that calcium remains in the cytoplasm, right? As long as you have that calcium there, it's going to go to the troponin, remove the tropomyosin, and you will continue to contract. So you're holding up a weight and you're contracting it. This is happening the whole time as you're holding this weight in your arm, for example. The calcium channels will have to close when the action potential ends. In other words, you're telling your muscle, I want to hold my arm out with a weight, and then I decide I don't want to hold it anymore. So that action potential ends. The calcium channels close. Now we have to mop up all that calcium that is in the cytoplasm and that you don't want anymore. So active transport is going to pump that leftover calcium and put it back into the sarcoplasmic reticulum. These DHP receptors are these voltage regulated receptors. So anytime voltage hits this channel, it's going to open it up. Here it's showing it with a little spring. It's not really a little spring, but any voltage that comes into the cell here will open that receptor. Let's stop right here, and we will continue on with the timing of excitation-contraction coupling in the next video.